This is our laboratory. I think that Alan has been there. He knows it's kind of like a chop shop for microscopes. We say, we pimp your microscope would be the... So this is an old microscope I salvaged. It's a 25-year-old microscope, and we put a $5 million worth of parts camera under it, much more extensive than anything on the Hubble. So this is 8K by 8K with 30% contrast at Nyquist for any engineers. So we acquire with this instrument, in an automated way, tomograms, one right after another, of sections that are 1/4 to 1 micron thick. And this is one example of a complete cell reconstructed with this 8K device. And you can see this is a saccular hair cell. And here, manually, there's been quite a bit of tedious delineation of structures. You can see how great this is if you go intracellularly and you begin to map these kinds of things. I won't go through all these movies, or I'll be here for longer than I'm allowed. Probably will be anyway. So in order to mark proteins, we need to get to where we can go beyond just recognizing proteins based on their structure. Here we can recognize actin. So we developed a technology, starting again some time ago, for using the generation of singlet oxygen, here with eosin, to paint molecules and deposit diaminobenzidine. We tested this with the acetylcholine receptor, with bungarotoxin and eosin as the singlet oxygen generator. Never published this. This is just a proof of principle for us. So this is purified acetylcholine receptor from torpedo. You can see the pore down the middle. Here it is in a tissue now. This is at a neuromuscular junction. So this proves that this technique will get us, the ability to recognize and stain things at that level. So we initially, because GFP won't generate singlet oxygen, Roger Chen's group developed, and then we perfected how to do this with a small domain that we cloned in that contained 4 cysteines and then ligands that bind to that. And this system, the so-called tetracysteine system, allows you to do dynamic imaging based on this ligand that's associated in the light microscope, and then chemical fix, expose it to stronger light in the presence of oxygen and diaminobenzidine, and you paint the molecule. Okay? And so here you see live gap junctions in this case, in the EM, and the structure that you see afterwards. Well, this was a great system. We did a lot of fun things with it over the last 6 to 9 years. We got it to the point, again unpublished, where we can see single protein complexes in synthesis in the endoplasmic reticulum, but still we weren't satisfied because these were hero experiments. So one of the postdocs in Roger's lab, Chai-Kun Xu, started working on a more powerful singlet oxygen generator. Again, we haven't published this. We're still waiting for the reports. This is called the mini singlet oxygen generator. It comes from Arabidopsis. Its singlet oxygen quantum yield is 0.47, so about 2 photons, 1 singlet oxygen. And you can see here, this is probably histone 2b, what we get to. Here it's driven to mitochondria with a mitochondrial targeting sequence. And here's the mitochondria that's been photoconverted after watching it in live cells. Here's another example, again, my favorite cell, the astrocyte. We're expressing this. This is now in mice after in utero electroporation. So this works in mice. It'll work in transgenics if you have a budget to make them. And here's, again, a slightly more interesting project to me, now looking at a neurodegenerative disease, or at least proteins associated with it. This is alpha-synuclein, looking at alpha-synuclein in long-term cultures, first in nerve terminals, then moving back to the cell body through multivesicular bodies, and then accumulating in the Golgi lysosomal complex. So we can begin to chart the systems that in this disease are susceptible. There are several disease genes in that system. Here's another system that I think we should apply this technique to, and that's, as I said earlier, mapping the nucleus. So here what we've done is we've made a histone 2b singlet oxygen generator, mini-SOG, and we've painted chromatin. Okay? And these are at about the same. This is a model by Janet Iwasa. But you can see that we can get down to the level and follow now in 3D with the techniques that I showed you a few minutes ago, chromatin structure. So there's one cell system that I think would be good. And talking with Sydney earlier, I think I have another idea. This is the Purkinje cell. If we want to understand the organization of the nucleus relative to the organization of cytoplasm, it's useful to have a cell where the dendritic arbor gives you a clocking of the orientation of the nucleus. Here's what I mean. So, of course, the Purkinje cell has this fan-shaped dendrite. If we keep track of the orientation of the fan, then not only do we know where the dendritic and the axonal arbors are, because they're almost opposite poles, but we can clock the rotation, and we can map nuclei with this technique. Not only the overall structure of the nucleus, but we can go in with specific new probes and look at specific genes. Okay? So this is, in the few minutes that I have, meant to summarize the whole brain catalog, so I'm going to jump back. I've not had a chance to show this movie in such a nice theater. With such nice acoustics. I paid quite a bit for the orchestra here. So no mice died in this. Can we get the lights down just a little bit? There we go. So Yuri, this is for you. This is looking at the dentate gyrus now. We picked some neurons to be represented in this. Which we knew to be the type that arose in adult animals. So this is from work with Nico Toni and Rusty Gage. We characterize these these granule cells as they appeared in the hippocampus. And so the cell that you'll see us come to in a moment, it's not an animator's fantasy, it's taken from the real anatomy. I use this movie to show not only the tyranny of spatial scales, but the tyranny of temporal scales that we deal with in our challenge to fill gaps. So how do we represent that? How do you have the equivalent of an oscilloscope with 2 time bases? Okay. So we chose with the orchestra that we hired to help us to do that by the composition, by the music. So you go from the kind of noise that you heard of the electrical activity to something slightly more delicate. As you ask, well, how are synapses made? And what's the time scale over which they occur? So here you see the fine phylopodial processes. The dendrites tend to send out, looking to hook up for a good time. They're looking for that hot axon in the street. Okay? And they're really not allowed to do that until my friend the astrocyte appears on the scene. Astrocyte, enter stage left. There you go. And the astrocyte is spritzing stuff in the vicinity, as Ben Barres and Eric Ulian have shown, thrombospondins and other molecules. And then they kiss and start to do interesting things, deposit receptors. And the otherwise cacophonous interaction becomes melodic. Is that how it happens, Yuri? That's right. And this is all deep in the brain and over again. And just making the movie is fun, but the real software, this is supposed to say funding to be continued. We're looking for funding. It's on YouTube, yeah. And I again have to thank Ted Waite and his generosity for helping with that. The actual site, I'll just take one more second here, is actually a lot of fun. You can go there. We'll have another release of the software at the neuroscience meeting. Again, it takes advantage of all of the infrastructure that the Allen organization has put together. This is an interactive environment and it's multi-scale, so we used an open source video game engine. One of the things that we wanted to do was allow the insertion of models of simulations that had been done on these sorts of things. So you go in and you play a simulation. So these are the growing neurons from the Gage lab from Brad Imeny's thesis, now placed appropriately in the hippocampus, getting imprinted on their pattern of activity and taking up their position. The other thing that you're able to do with this site is you can bring in a cursor in 3D. I think that's going to happen here in a second. I'm a little faster than my movie today. So this is the 2.5D environment. I want to make this work in a 3D environment, really 3D soon. But you right-click on this, and you can ask from the ABA, you see, spatial query ABA, give me all the genes in the place I just put that. And then you bring up another page from the Allen site and lets you, so this is one of many sites that we've linked in. So this is, how would you say, an interactive window. Not only can you do this, but you're meant to be able to bring your data in, kind of like the Street View model that I started with. And you can go to the protein data bank and paint and decorate with channels, et cetera, et cetera. So I think with that, I'll just acknowledge that I raise money and I'm a spokesperson, and everybody else does the work, and that because of the characteristics of this theater, I needed to say that we're really into the future. Thank you.